Hi, it's Ramon Matawarren for my BMI224 project. I chose TelaprevirIL and IL28B for my drug gene pair. IL28B gene is also referred to as interferon lambda 3. I thought I would contextualize the presentation by presenting a brief overview of the hepatitis C genome and the life cycle of hep C virus. Hepatitis C virus has many genotypes, and genotype 1 is the most common in the US and Europe. The hepatitis C genome consists of a single open reading frame, and that is then translated to produce a single protein product, which is further processed to produce smaller active proteins. The structural proteins in light blue shade include the core and envelope proteins, and the rest are the non-structural proteins. I'd like for you to pay close attention to the notation at the bottom of the slide, as this clearly reveals the targets for the new drugs against hep C, especially the serum protease enzyme, which is the subject of this topic. This is a basic overview of the hepatitis C virus life cycle. When the virus enters the liver cell, it releases its RNA and is translated into a polyprotein containing structural and non-structural regions. The polyprotein is processed by proteases into several polypeptides with different functional roles in their virus life cycle. As alluded to in the prior slide, the initial cleavage of the polyprotein is performed by the NS3-NS4 protease, which is a target for the drug telaprevir. This enzyme is highly conserved across most strains, without which the hepatitis C virus life cycle cannot proceed. Moving on, the virus is replicated with the help of a polymerase enzyme and then assembled, transported, and released from the cell, as it's shown on the upper right-hand corner of this particular slide. Telaprevir is marketed under the brand name Incivek and is also available as Incivo in other countries, and was developed by Vertex Pharmaceuticals and Johnson & Johnson. It is available in 375 milligram tablets, usually taken 3 tablets twice a day, 12 hours apart, with a high-fat meal, and is used in conjunction with PEG interferon and ribavirin. The cost of telaprevir-based triple therapy is estimated at $189,000 per sustained virological response, which means no detectable viral load after 6 months of treatment. The toxicities of this medication include anemia, which led to discontinuation of treatment in 3% of the patients, and rash and itching, which led to the discontinuation of treatment in 6% of the patients, although there were no fatalities. A landmark genome-wide association study in 2009 involving 1,600 chronic hep C patients identified the SNP around the gene coding for IL28B. The IL28B polymorphism, RS12979860, was found to be the single most important predictor of sustained virological response. The SNP on chromosome 19 is located upstream of the IL28B gene encoding for interferon lambda 3. It is postulated that the interferon lambda may produce a better immunologic profile that could favor viral clearance. It has been recently demonstrated that the expression of hepatic interferon-stimulated genes, or ISG, is strongly associated with both treatment response and the genotype of the IL28B. This table shows that the more favorable genotype variant CC was more common in Asians and those of European ancestry. Genotype should, however, not be used to withhold hepatitis C virus treatment, since those with the less favorable genotypes can also be cured. This clearly shows the advantage of CC genotype over others in inducing sustained virological response. There is already a pharmacogenomic labeling available for telaprevir for both the US and European market. The pharmacogenomic information is limited to clinical response variability according to the genotype. The IL28B genotype will likely be incorporated with other clinical variables to help predict response and help personalize therapy before commencing treatment. Those with favorable genotypes may require a shorter duration of treatment, and those with unfavorable genotypes are more likely to benefit from the longer duration of treatment, along with the addition of the direct-acting antiviral drugs such as TelaprevirIL or the more recent iteration. These are my references, and thank you for your attention.